Hello, I am Irene Evnukidou and I work as a researcher at Dr. Savian's group at INSERM at the Center of Research on Inflammation in Paris. We are interested in understanding what mechanisms the organism uses to resist to pathogens, cancer and autoimmunity. Our organism has to confront every day various threats for our health. Our immune system can protect us against all these threats with various defense lines. One of the most important means for our immune defense is the recognition of unhealthy cells, such as cancer cells, by cells of our immune system and thereafter their elimination. In order to accomplish this elimination, the T cells of the immune system must go through a series of steps in order to get activated. First, their Tcell receptor should recognize antigens, small pieces of proteins that are presented at the cell surface of the unhealthy cells. Once this interaction is established, a complex pathway of interactions between proteins inside the T cell, called signaling, will allow T cell activation. The activated T cell will now be able to eliminate the unhealthy cell. We were interested in understanding what is the fate of the T-cell receptor after internalization, since for a long time this was considered just to end signaling. We identified a molecule, IRAP, which is known to play an important role in other immune cells, but had never been studied before in T-cells. IRAP characterized the vesicles that contain the T-cell receptor inside the cell. In this picture, you can see a T-cell. The vesicles in yellow contain both IRAP and the T-cell receptor. We showed that IRAP interacts with molecules important for the correct function of T cells. In the absence of IRAP, TCR localization changes, with most of the TCR now found at the cell membrane. This leads to defective TCR signaling and reduced recruitment of signaling proteins to the immune synapse formed with the unhealthy cells. You can see an example of an immune synapse in this picture, where the unhealthy cell is blue and the T cell is green. The recruitment of a signaling molecule to the immune synapse appears as a clean red line in control cells, which you cannot see anymore in the absence of IRAP. A major function of T cells, which is IL-2 cytokine secretion, that allows further activation of the T cells, is also affected. Interestingly, we found that there is TCR signaling inside the cell, which disappears in the absence of IRAP. We showed that this endosomal signaling is very important for correct T cell function, especially in the case of weak antigens that need to be at high concentrations to be efficiently bound by the T cell receptor. All these defects result in defective T cell response and inability to control tumor development, as we showed in a mouse tumor model. Mice with T cells deficient for IRAP developed much bigger tumors and demonstrated very low response of T cells inside the tumor. Therefore, we propose that T cell receptor endosomal signaling is essential for T cell responses. Our results allow a better understanding of the mechanisms of T cell activation and could be used to improve current immunotherapy tools against cancer. To go further in our understanding of the role of IRAP in T cell activation, we are currently studying the detailed composition of the cellular compartments that form the signaling pathway of the T cell receptor. Thank you for watching our video, and I would like to thank INFLAMEX for allowing us to present our study. We invite you to follow INFLAMEX News to discover other studies presented by their authors.